So my name's Matt, and I'm based at University Hospital Southampton. Like that? So I'm based at University Hospital Southampton, and we've just received funding from Sarcoma UK and GIST Cancer UK, which has allowed me to integrate with Zoe and Jack's research and start to look at natural killer cell therapy. So thank you very much for funding our work, and really excited to show you what we've been up to over the last few weeks. So my lab all focus on the immune system, and we're really keen on activating the immune system against cancer. So just a very brief overview of what we're talking about here. So our immune system's really designed to protect us from pathogens. So if any bacteria or viruses enter our bodies, the immune system can react and detect that and then destroy that. I'll go back. There's a whole range of different cells, so called innate and adaptive immunity. And we're working on a particular cell type called a natural killer cell, NK cell, which is shown somewhere on that image, which I cannot see now. Oh, there we go, in green. Don't know if I've got a pointer. And it really works in a sort of like a real tight balance. So our immune system has to be able to respond quickly and effectively to destroy pathogens. But if you get too much immune activation, it can lead to things like autoimmune disorders. So that can be too much inflammation is also bad. So it has to be really tightly regulated, but able still to do its job. And so I currently lead a team at the University of Southampton, which is all focused on natural killer cells. And we love this cell type, and hopefully by the end you'll like them too. And what they do is they basically go around our body, sensing all of our cells, and they'll sort of interact and touch with them and think, is this a good cell or a bad cell? Do I need to get rid of that? And it's their job to identify rapidly any cells which are abnormal, maybe have got mutations which should be destroyed, or have become infected. And natural killer cells are really good at that. And these guys have been around for hundreds of millions of years. So you can track these back even to things like frogs. So these have been around protecting us throughout our evolution for hundreds of millions of years. And what they do is they use activating receptors, which I've got shown in green, and inhibitory receptors shown in red. And it's really this really tight balance between, is there too much activating signals here? I'm going to kill that cell. If there's lose the inhibitory signals, they can also become activated. And healthy cells have a lot of signals for those inhibitory receptors, and basically just say, don't kill me, I'm the good guy. When they do sense a bad cell, they secrete these granules, which are shown sort of in purple here, and they basically get secreted from the natural killer cell. And my 6-year-old son likes to think of it as a gun, and they sort of basically punch holes in those cancer cells, and they'll literally form a pore in the cell, and that will lyse that cell and cause it to die really rapidly. And the good thing about natural killer cells is they're not specific to any certain pathogen or cancer type. So in the lab, we take healthy donor natural killer cells, and we can put them in front of a breast cancer cell line, a GIST cancer cell line, or a leukemia cell line, and the same natural killer cell can kill all of those really quickly. And we see death of those cancer cells within minutes in the lab. That's a really, really quick effect. Another important role that natural killer cells have is actually supporting other immune cells. So they're not just targeting and destroying their target cells. They can also recruit and bring in other immune cells, which are important for anti-cancer immunity, like the ones Jack spoke about earlier. I've included a video in my talk, and this is the first time I've ever given a talk with a video, because there's always technical problems. And there is, because I can't press play. Oh, here we go. So you can see the natural killer cells. So these are the small cells here. And then this is a single cancer cell here, labeled green. And now you've got adverts on YouTube. But you can see what happens. If we play it again, maybe, if that's okay. Maybe. Basically, you can see the natural killer cells take a little while to recognize it, and they're feeling those receptors on the outside. So these are the natural killer cells. They're really small, and they're basically into contact with that. And then when they realize this is a bad cell, we need to get rid of this. And they cause it to literally lyse and burst. And you can see that happens really, really quickly. And the good thing about natural killer cells is that's not the end of the story. So they don't die themselves. What they do is they pack their cells in really tight, and it stops those cytotoxic granules killing their own cells, because that would be like one hit, that's it, their cells are done. But they can go on and kill multiple different cells. There's multiple targeting of cancer cells with the same natural killer cell.
What happens to the, it bursts, what happens to the stuff then that's released?
It will get taken up by, say, macrophages, and they'll just it will get cleared from the body. You can get something. So I come from a leukemia field, and you can get something called tumor lysis syndrome, which is too many cancer cells die too quickly, and that can cause problems. But natural killer cells don't do that. Thank you for that.
I'm glad you're doing this.
And not me. So the natural killer cells are very small, but they've got a lot of power. And importantly, they're already in clinical trials for patients. So what we can do is we can take these natural killer cells from a healthy donor or even from cord blood, and you can then build these up in the lab. So you make them expand and proliferate. So there's billions and billions of them. And then you can then infuse them into patients, and then they can go off and then have their effect. And that's already in clinical trials at the moment for other types of cancer. And I'll come on to that data a little bit later. And it's also, I've got here off-the-shelf use, and that is literally off the shelf. So what happens in Southampton at the moment is patients with lymphoma are treated with a CAR T cell therapy. So that's a T cell, which is a different cell to an NK cell. And they give blood. The T cells are shipped off to the Netherlands. They're re-engineered to target the cancer cells, and then shipped back to Southampton and then infused into the patient. So that takes a number of weeks. It's obviously really expensive, and it's a personalized product. So that's one patient, one treatment, one injection. But what NK cells can do, because they're extremely, extremely safe, you can make them from, say, one healthy donor, make trillions of these cells up in the lab, and then they're literally off the shelf, so we store them in the freezer. And that same NK cell product from one person can be used to treat 10 people, or it can be used to give into the same patient 5 different times. So that obviously makes this a lot cheaper, and it reduces the production time. But also importantly, you don't have to wait 3 weeks for that treatment to come back to you. You can just take it out of the freezer at the hospital and give it to someone straight away. So that's why there's so much excitement around using NK cell therapy at the moment. So previous research has shown that NK cells are associated with reduced relapse rates in GIST. So the black line here is high NKp46, which is a receptor expressed on NK cells, and they've looked at low and high levels of this NK cell marker. And you can see that for patients who had more NK cells within the tumours, that was associated with sort of better prognosis. And also during imatinib treatment, it's known that after imatinib treatment, so this is around a year, I think, afterwards, after imatinib treatment, the NK cell numbers increased within the patients, both localized and metastatic tumours. So we think there's a real opportunity, because we know natural killer cells are present within the tumours, but they're obviously not doing their job properly, and they're not working as they should do and can do. So what our project is all about, and what with your funding we're able to look at, is can we sort of wake these NK cells back up and make them target the cancer cells as effectively as they should be able to. So coming back onto how NK cells work. So this is an inhibitory receptor, and it binds to ligands on the healthy cell, and that switches off that natural killer cell, and there's sort of low levels of activating signals. And for a natural killer cell to become activated, you can get more of the activating stimulus, and this is quite often when a cell becomes stressed, it shows more of these on its surface, and that's like a danger signal to the immune system to eradicate that cell. In addition, you can get loss of, so this is MHC class 1 here, loss of these inhibitory signals, and then the NK cell is not inhibited. And NK cells are really, the inhibitory receptors dominate their actions, so if there's any inhibitory signals there, it will just switch them off. So it's really like a safety first approach for the natural killer cells. So what we can do is we work a lot on a receptor called NKG2A. So this is one of these inhibitory receptors. So there's a whole big range of different receptors that these NK cells express. So one of them that we're particularly interested in and been working on recently is called NKG2A. And it binds on a healthy cell as well as on cancer cells to something called HLA-E. And they interact together, and that then inhibits the natural killer cell. So healthy cells have this, but actually a lot of cancer cells actually increase the expression of this HLA-E protein on their surface, and it kind of acts like a shield. Because if they have way more of that than a normal cell, it just turns off the immune system as soon as they come into contact with it, and then they won't then lyse it. And my team, so we've just published a few weeks ago, so Lara is a PhD student in my team, and we just published this really recently. This is actually for lymphoma. So I think I mentioned at the start, all of my team work on lymphoma and leukemia until we receive the Just Cancer UK funding. But this is just to demonstrate sort of the power of blocking these inhibitory signals. So this is an antibody which comes in called NKG2A blocking antibody, monalizumab, and it can basically get in the way of this interaction, and then it stops that inhibitory signal go into the natural killer cell and turning it off. So you can see with lymphoma cells, these are the percent of these cancer cells that were lysed at this time point. And you can see with here in black, average around 20% of the cancer cells had died at the time point we looked at them. But if we were to block NKG2A with monalizumab here, which is a drug that interferes with this process, you can see that some of these are going from only 5% dead to 30 or 40% dead at this time point. So if we left this for longer, this is going to, you'll kill greater percentages of this. It's just a snapshot in time. But you can see we're more than doubling the ability of NK cells to work against cancer cells when we interrupt these signals. And what's really important, as Zoe mentioned, was drug repurposing. And this NKG2A blocking antibody called monalizumab is already in phase 3 clinical trials for lung cancer. So anything we find which is exciting with drugs like this can be really quickly repurposed for clinical trials for GIST. And another project that we're able to look at with this funding is something called chimeric antigen receptor NK cells. So they're already CAR T cells, and they're the T cells that I mentioned earlier that are given to lymphoma patients on the NHS, where they reprogram them to bind to cancer cells. So the chimeric antigen receptor, or CAR, is this shown in green, and it basically allows immune cells to recognize the cancer cells much better and to stick to them. And then this is an activating signal. So when that binds to the cancer cell, it's a really strong activation signal and tells them to kill that target cell. And you can see that's the lysis of the cells. But CAR NK cells have also been developed. So similar to the CAR T cells already approved, CAR NK cells are now in clinical trials. We've spoken about AI already and how we can use that for science. So I tried to use AI to make a figure of this for this talk. It didn't quite work out how I was hoping. But you get the idea. And this is clinical trial data published last year from America by someone called Katie Rezvani. And she used these CAR NK cells to treat lymphoma and leukemia patients. So CLL is a type of chronic lymphocytic leukemia. And then there's different types of lymphoma. And I won't go into too much detail, but basically the dark blue is CR, which is complete remission. So at this time point, they couldn't detect any of the cancer left in the patients. And you can see that in the top row, 6 patients, 5 out of 6 had completely eradicated the tumour. And there's different rates for different types of tumours that they looked at. But this just shows the power of this. And this is all from a single infusion of these CAR NK cells. So one infusion. And these are all taken from cord blood. So it's not the patient's own cells that may be dysfunctional because they've been suffering from cancer or cancer treatment. They're fresh. They're from a different healthy donor. So they're able to act really well. And what we want to do is translate this now into GIST. And I think what's really important is the excellent safety profile. So in this clinical trial, there were no severe toxicities found. So this is obviously better than things like chemotherapy, but it's also better than the CAR T cells that I mentioned that are already approved on the NHS. So they actually can work too well, and they can induce too much inflammation, so things like cytokine release syndrome. But actually you don't see that with natural killer cells, and that's because they have all these inhibitory receptors, and they don't become too activated. They just do their job, but they don't cause too much inflammation to cause too much toxicities. So I think they're really exciting and hold a lot of potential. And as I mentioned, the reduced cost and production times. I think if we can get them to show that they're effective, they're not going to be expensive for the NHS, which is really important. So I mentioned with Sarcoma UK and Just Cancer UK's support, we've just been able to start a project. So my team were looking at lymphoma and leukemia, and they still are, so I have a big team looking at that. But now with this funding, which just started a few weeks ago, we're able to develop all the tools that we've developed for lymphoma and leukemia to now look at GIST, which we're really excited to do, and have integrated with Zoe's team and with Jack as well. The aims of the project really were to look at NK cell frequency, phenotype, and spatial location. So how good are the NK cells that are within the GIST tumours? Are they able to function? Can we wake them up or turn off some of these inhibitory signals to make them work better? And that's the immune checkpoint. So that is those inhibitory receptors that I mentioned earlier. So can we switch some of these off and try and reawaken the natural killer cells against the cancer cells? But in addition, we want to develop the first CAR-NK cell approach for GIST. So that will be taking healthy donor NK cells, expanding them up in the lab, and then putting them onto the GIST tumor models and seeing if we can better eradicate them that way. So we've got a multi-pronged approach using patients' own NK cells and using healthy donor NK cells. And I'm really excited to see how that develops. I said we've integrated with Zoe and Jack's research. So this is, Jack has already shown some of this. This is some of his single cell analysis of NK cells with just patient tumor samples. So basically he's shown that natural killer cells are there, and it's just now looking at how good are they going to be if we can find a way to turn them back on, or do we need to give ones from healthy donors? And Jack has already shown this picture, but here it's just with the yellow dots, which are the natural killer cells. So now Jack can look exactly where the NK cells are within the tumours. And are they actually at a site that they're going to be able to do any good? Or are they excluded and we've got to find a way to let them enter? So with this funding that we've just received, so Dan is pictured on the left, and he just started working on GIST a couple of weeks ago in my lab. And he's a really nice guy and really hard-working person, so it's great to have him join the team. And what he's been using is flow cytometry, which is a different type of technique, and it basically passes the cells through a small amount of liquid, and the machine is able to look at each cell really quickly, individually. So every dot is a cell on there. And then what we can do is we can look at those cells and use, and then basically check what's on the surface of those cells. So this is something called PD-L1, and that's called HLA-E. So that was the molecule on the surface of the tumor cells that turned off the immune system. And PD-L1 does the same thing, so it turns off the immune system, but it can also switch off T cells. And what we've been looking at is how imatinib can affect the expression of these molecules. And also how interferon gamma can change the expression of these molecules. And this is important because as soon as the natural killer cells become activated, or T cells become activated, you get this interferon gamma secretion, which is a cytokine. So it's a protein designed to have anti-cancer properties, and it can stimulate the immune system. But what we found in GIST is that it can also, sort of paradoxically, turn off the immune system. So you can see here with interferon gamma, so this is the GIST tumor cells in a dish. This is them with interferon gamma mimicking an immune response. And you can see that PD-L1 goes up a lot, and so does HLA-E. So it goes from around 800 to nearly 3,000. So there's a massive increase. So you can see as soon as you get some immune cell activation, any immune cell activation, within hours, you get upregulation of these proteins on the GIST tumor cells that are acting like a brake and turning the immune system back off. So that's obviously a problem that we want to be able to overcome. And what Jack has shown is that imatinib actually, if that's there at the same time, it can prevent that upregulation. So you can see HLA is much lower, and so is PD-L1. So what we think is that maybe imatinib could interfere with this sort of brakes on the immune system and unleash the immune system. So potentially we could use imatinib to combine it with natural killer cell therapy and make it work even better. And this is just to show that. So this high HLA-E you can see is here, and then this goes up even more as soon as the immune system is activated against the cancer cells. But then imatinib can actually reduce that and may be able to restore the NK activation. So what Dan's going to be doing over the next few weeks is then combining natural killer cells in the lab with the GIST cells under these conditions and seeing if that is going to promote natural killer cell activation. And potentially we could combine this, say, with a CAR-NK cell approach to make sort of prime those tumours to make them have an even better response. That was my final slide. That's what Dan's been up to with the last few weeks of your funding. So thank you very much, and thanks for listening.